Micros, Chairman of Commission C, Federal Institute of Drugs and Medical Devices, Germany, and Dr. D.S. Lohar, former Director, Homoeopathic Pharmacopoeia Laboratory, India. And there are three speakers for this session, and with the permission of the chair, we would like to start with the session, sir. Please.
Yes, dear ladies and gentlemen, I hope you find back to your seats after the short tea pause. And as you may have noticed, we are a bit short in time. So I can announce that we will try to get the program of the morning ready before lunch. So everything will happen in the morning, but the discussion will be a bit shorter in the end. So it is my pleasure to announce here that we will hear the first talk from Christian Moll. Christian Moll, you heard him yesterday. He is General Secretary of ECHAM, the European Coalition of Homeopathic and Anthroposophic Medicine Producers. And he has an interesting career because he is a pharmacist. Christian was educated in homeopathy and in anthroposophic medicine, but as well, he holds a master in management of non-profit organizations. So that's why always his talks are a bit strategic and give you backgrounds. And just to make clear for all of us, we had now the status quo. Now we are dealing with strategies, and then we will go to future options and future innovations. So please, Christian, the floor is yours.
Thank you. Thank you, Chairman. Again, good morning to all of you, homeopathic community here together in Delhi for this brilliant event. I would like to speak today on strategic aspects to be considered with regard to this quantification topic. I call it a little bit broader quantification, meaning assay limit test or anything that can help to detect specific substances. I want to remind what my presentation of yesterday, just a general principle of it, that if we ask ourselves what is a standard is actually something that is developed between state actors, market actors, and the demand side. So it is something that actually influences all three most critically, and the Pharmacopoeia in particular has the task to protect public health, to grant a high quality, and has an impact on all of these players, state, market actors, and the demand side. And yeah, what are possible fallacies again? There could be fallacies to develop standards for homeopathic preparations if they are too low. No proof of constant quality is possible. The safety and reliability of the product may be questionable. This would be when the standards are between hyphens too low. I don't specify what the word low shall mean in detail. On the contrary, they may be too high if they're just copied. And this is which would run under the title isomorphism. You copy standards without a proper reflection, or with a reflection that can be discussed. And the consequence could be that you have a market failure or a problem with the availability, in particular for the rarely used substances in homeopathy, and then that you favor a monopolist industry in this triangle, that you have a major impact on the market actor side with a hypertrophy of one or two actors. So in this issue, keeping the balance in the triangle of tensions, it is my presentation just highlights on saying, well, stick to the essential, stick to the meaningful, and legitimacy is a core issue. And legitimacy is achieved, as I showed yesterday, if a thorough discussion takes place amongst the experts where the points of view are gathered. And this is practice in the Pharmacopoeia committees we've heard this morning. And on this issue, however, I think there is a need for more discussion. So I will not repeat this. It has been said several times that the definition of the European Union of homeopathic medicinal products bases on manufacturing, and this produces a multi-substance mixture. So a question can be raised. So if we speak about plants, for example, yeah. The question can be, well, when is quantification, such as assays, limit tests, when is that really meaningful, essential? And I would propose an idea, a strain of thought that may contribute to this discussion. So we've heard the differences of the pharmacopoeias today already, the different approach in the French pharmacopoeia, the different approach in the German pharmacopoeia. The European law is apparently a little bit unclear because we still have these different approaches. So I'll not go into that. We have a technical guide of the European Pharmacopoeia that gives statements. It introduces the principle of assaying. It uses wordings like where appropriate, where necessary. It somehow reflects the need of this to introduce some quantifying aspect, but it is not, let's say, so mandatorily clearly expressed in the wording where necessary, where appropriate. And this, of course, can be challenged. There may be different points of view on this. If we look at the regulatory practice, there are also obvious differences. In Germany, the Netherlands, and Italy, for example, dossiers are accepted without quantification of non-toxic substances. We've seen that the toxic substances, there is more consensus, but the non-toxic substances, you may have dossiers without quantification. Whereas France, we've heard that, always requires the detection, the quantitative detection of marker analytics. So other just indicators of the ongoing discussion, a publication by Professor Keusgen, a publication by Professor Fouraster and Bianca Maria Bruno, a publication from India, very interesting, published in Homoeopathy 2015 of Yadhav and his working group on marker analytics of on biological activity. We've heard that earlier this morning, that this is also an interesting trace. And just a glance at traditional Chinese medicine, publication 2009. There is an ongoing discussion on this topic as well in TCM. So it's not only a topic that is dealt with in homeopathy. So thorough analysis is needed. This would be my motto, a thorough analysis, and we are doing the best we can as association with 45 members about from 18 member states, including French and German companies. We are doing the very best to contribute to progress on this discussion. So what is really meaningful? What is really, what is adding value? Because we want a high value in our sector. What is really added value? So, we've heard this also yesterday. I mean, governance of medicines departs from efficacy, safety, and quality. So we, in our association, we thought that we have to somehow look at these three pillars of pharmaceutical governance and try to streamline quantification thoughts with those principles. So, the first one, the criteria efficacy. We've seen that the example relates to botanicals. So the overall logic of quantifying substances in view of efficacy is that you want to guarantee the presence of your wanted substances. You want them present, they have to be present, otherwise the medicine is of bad quality. And this is usually mostly the dosage efficacy ratio. And this mainly bases on the direct action of substances at receptor level. You know, the receptor model of allopathic medicine, an antibiotic to kill a target bacteria as a very simple example. Whereas now, from the efficacy point of view, the homeopathic principle of action is regulation, stimulation of a whole system. It's not a molecule on a receptor. It's the stimulation on a whole organism, according to the Similia principle. And an example here, chamomilla, may be recommended in different potencies for different pictures, amongst others, fever, teething, inflammation, and cramps, according to what the prescriber, what the recommender, the pharmacist, sees. So my conclusion would be, the conclusion that we draw in our working group in ECHAMP is that assays cannot reflect direct pharmacological action of homeopathic preparation, such as we aim for in homeopathy. So efficacy does not deliver a rational basis to require assays. This can be discussed. This was our interim result. Safety, the second pillar, safety. The encompassing rationale for safety is that toxicologically relevant substances be controlled. You have to know how much is in there. And you don't want more than So it's absolutely clear that you need to do an upper limit of your toxicological substances, and this must be a very well done. So from a safety perspective, what is really clear is that an upper limit needs to be set for toxicologically relevant substances, and this is with some smaller variations on the topic, general agreement. Now, quality. We found that the encompassing rationale for quality is that a high-quality preparation must be obtained by a high-quality starting material. So any dilution you make, at the beginning there must be a high-quality starting material. So I have the example now of Geum urbanum, plant of the family of the rose family, which is very typical, the tannic atmosphere, let's say, of the plant, astringent on proteins. So can we say that Geum urbanum without tannin compounds would be a good starting material? I would say no. If we have Geum urbanum without tannic compounds, you have a strange Geum urbanum. So, interim conclusion, from a quality perspective, high quality shall mean that a starting material, our batch of Geum urbanum, is a proper specimen of the species. Geum urbanum produces tannic compounds. I want to find them in my batch. So, relevant compounds should be detected. Tannin compounds should be detected for Geum urbanum. So, now the next question is, well, how to do that? With what technology? How far? Remembering the triangle, I'm setting a standard in a complex environment. The interim answer we give as ECHAMP, our experts in this very lively discussion, is that TLC or HPTLC are valuable tools that allow identity and detection. I'll give an example of Geum urbanum thin layer chromatography processed by a method of the HAB. And you see, this is Geum urbanum, and you see this chromatogram. So, this is not a general plaidoyer for thin layer chromatography, but it shows that thin layer chromatography, TLC, HPTLC, they are valuable tools. And if we look at the, I know, sorry, this is German, Gallussäure, gallic acid, that's a tannin compound, you see very clearly that the reference substance can clearly detect spots at that height. So, I would like to draw a wrap up. We've seen efficacy, quantification basing on efficacy. That's difficult. It can be discussed. We found that it's mostly difficult to deduct that really from the efficacy principle. Safety, we've seen absolutely clear upper limit. That's very clear. And now the quality part, I'd like to summarize. Homeopathic preparations are defined by their manufacturing method. They are multi-substance mixtures. They must be of high quality. A high-quality preparation can only be obtained from a high-quality starting material. High quality shall mean that a starting material is a proper specimen of its species. Think of an untoxic belladonna. What do you do with an untoxic belladonna? No way to use that. And this is why we say that typical characteristic substances for the species, what is the atmosphere of the species, and sometimes it's very clear, it's around substance groups, sometimes it's a molecule, or mostly it's substance groups, and that TLC and HPTLC are meaningful tools to detect the presence of meaningful compounds in a lot of cases. And a semi-quantitative detection is possible. Bayut Danyavad.
Yes, thank you, Christian, for this interesting talk. We will immediately proceed to the next talk due to our shortness of time. And this is a premiere now, because you know you can play tennis single or double. And now we will have a double talk, which is given by Todd A. Hoover, who I learned is qualified in conventional medicine, emergency medicine, occupational medicine, but as well in homeopathy. And he is somebody who has a degree in yoga, and he wrote a textbook on Ayurveda. So if we near ourselves to the paradigms of integrative medicine, he is the perfect example how to integrate lots of different approaches in one person. And Robert van Hazelen will overtake during the talk for the second half, and I don't have to introduce him because you all know him.
Thank you. And I want to thank Dr. Manchanda for working with Robert, both of them for creating this Congress. It's been very good for me to see, the whole landscape of the different pharmacopoeias, regulators, et cetera, and Ayush for supporting the effort. It's been very good so far, and I'm sure it will continue. I want to start with our first slide, and it says, respecting homeopathic standards while modernizing monograph evaluation, and not to lose the baby with the bathwater. And this is an expression we use to say, yeah, it's good to modernize, but we don't want to lose the homeopathy in the process. And I've heard this echoed through a number of talks, and I stopped keeping track, but Dr. Renaud mentioned it, Dr. Honda, Dr. Manchanda, different people mentioned the same kind of idea that we need to modernize, but we need to keep the homeopathic principles intact. So far, we've heard over and again that quality, safety, and efficacy are the pillars of drug evaluation and monograph evaluation. So far, we've heard a lot about quality, a lot about safety, and not so much about efficacy. So I'm going to focus our talk really around efficacy. The HPCUS, as you know, has a unique role in performing some, not regulatory function, but approval function for drugs that come into the United States. And therefore, we're charged with looking at both safety and effectiveness or efficacy of new drugs. The question is, how do you do that? And for this talk, I want to mention that we're really looking at single ingredient drugs, not combination products. So here's our objectives. We want to differentiate what are the allopathic and what are the homeopathic pre-approval needs, how have we evolved our proving standards over the last 5 or 6 years to produce the standard which was published in 2013, and where are we going with our clinical evidence standard, which is being produced at this time over the last 3 or 4 years, and what are the future needs in pre-market assessment. So, I hope I didn't skip a slide there. In 2013, as I mentioned, we worked on, I think I did skip a slide there. Yeah, okay. So just to begin with, allopathic drugs in the United States are approved through the CEDAR, which is a subdivision of the FDA, and over-the-counter products are monographed through the United States Pharmacopoeia. Homeopathic drugs, as you know, come through the Homeopathic Pharmacopoeia, or the HPCUS. They have very different approaches in how they look at efficacy. In allopathic medicines, we're really looking for a single diagnostic indication, whereas in homeopathic drugs, what we've come to is we're looking for a clinical composite picture. So to put that simply, if a drug's used for high blood pressure, all we're interested in allopathically is, does it lower the blood pressure in the majority of people? In homeopathy, we might have a drug that's used for high blood pressure, but we're interested in that particular subsection of patients, for example, nux vomica. Does it work for patients who need nux vomica who may happen to have hypertension as part of their case? So we don't look at them in the same way, and they shouldn't probably be measured in efficacy in the same way. In 2013, we started looking at, well, what kind of evidence do we need to determine what a clinical picture for a given monograph looks like? And we went to, historically, provings have been done to do that through drug discovery. And it's a unique tool in homeopathy. It's not well accepted in the allopathic community and by regulatory agencies, especially in the United States, by the FTC or by FDA. They really don't look at provings in the way that we do in terms of acceptability. The LMHI and the ECH harmonized their guidelines, and most, I would say most, if not all of the changes we made in 2013 were adopted by those organizations in 2014. How did we go about this? We looked at things historically, what was done in provings, what has been done over time in provings. One of the members of our group has done something like 80 provings himself, so we had a lot of expertise in the panel that were developing these. We looked at what are the current scientific community standards. Interestingly, homeopathic doctors were some of the first people, or probably the first people, to systematically incorporate things like placebo controls, blinding in studies. The AIH adopted these kind of standards, I think it was in 1885. So it really didn't enter the allopathic thinking until probably the 1960s in an organized way. Regulatory authorities, what were the demands of the FDA and other kind of regulators? How do we need to meet those standards? ICH guidelines were considered in our thinking process. And what was available in the current research on methodologies? Were placebos used, the process of provings, not how were symptoms taken in provings? Many of the different aspects of how provings are conducted we considered. This is a basic overview of how we look at monographs. Quality is in the first couple of steps. Safety is in the middle kind of steps there. And efficacy really is in looking at proving submission and clinical data. All of this goes to the monograph review process, and we either approve or reject the monograph. This is done by two main committees. The Monograph Review Committee on the left mostly handles quality control and safety issues. The Clinical Review Committee on the right mostly looks at safety in terms of first use in humans or for new products, as well as the provings or clinical evidence for efficacy. And this is composed primarily of physicians and epidemiologists. So where are we going with things? As we've produced the proving guidelines, we've probably looked at 10 or 11 new provings now for monograph, and we've been kind of using the physician, the Pharmacy Revision Committee as our instrument to ongoingly look at how provings are conducted and can we alter our guidelines to actually make them better over time. So far, they look pretty good. So I'm happy with what we've created. The next step is something I'll talk a little bit about today, is we've looked at, well, how do we know that provings are actually valid? This is a question that's not often asked in homeopathy. Well, of course they're valid. They've been around for hundreds of years. We have all this clinical evidence which shows that they're valid, but no one's actually looked at assessing this. So we're starting a pilot project. We're actually about halfway through it, which we'll present at the next LIGA meeting, as well as the HRI, which investigates validity of provings. The next is something Dr. Van Hasselen or Robert will talk about, case reporting standards, which we're working on, and observation clinical trial standards as they relate to homeopathy. So the research project that the HPCUS is funding has asked the question, are provings predictive of clinical outcomes? In other words, do the symptoms that come out of a proving, when they're used clinically, do they actually result in patients that get better? It's a pretty simple question. So you have two sets of symptoms. We have symptoms from provings, and we're looking at both older provings, ones conducted by Hahnemann, as well as modern proving in this particular research. And then we're looking at cases where symptoms were used and seeing where those intersect. And then the cases that we're using actually were produced by the Vitulcas Compass people within their database. So we have hundreds of cases that we're using combined with the symptoms that were derived from the provings. Where those overlap, we then compare to see what were the outcomes, and it's a simple relational study. So basically, this study, what we're hoping it will show is that either the symptoms for provings are predictive of outcomes or they're non-predictive. And we don't actually know the answer to the question yet. So by measuring the outcomes themselves, we can see that those symptoms that were chosen for provings led to the result or they didn't. So I'm going to hand off the talk to Dr. Van Hasselen, who's going to talk more about the clinical aspects of the monograph process.
Thank you.
Yeah, so when about 7 or 8 years ago, we started within the HPCUS in a working group to look at all these guidelines, we realized that the clinical data, the effectiveness part, is really a very, very complex issue. And so we sort of said, well, what do you do with a complex problem? You break it down. So what we did is we said we're first going to focus on the proving part. And so in 2013, the proving guidelines were published. And this presentation up to this point reflects, I think, some of the results that have been achieved, and I think which are very encouraging, and some of the ongoing work in that domain. So my part of the presentation will focus on the other clinical data part of this story. And this is what we've been working on, I would say, for the last 3 or 4 years in the same working group to look at all the other clinical data. So in terms of the kind of clinical evidence, we've broken it down into 3 large sections. Case data, observational studies, and clinical trials. And without going too much into the detail, these different designs have different strengths. You know, case data are very externally valid because they represent the clinical reality, but they also have limitations. And so different methods have different strengths. So we've worked quite hard on the clinical data part. We're planning to publish the clinical evidence guidelines in 2017, so this year. And it will include preferred types of clinical evidence, acceptable methodologies, levels of clinical evidence sufficiency, and reporting standards. So this is really, this year we hope to publish the fruits of a lot of hard work in that respect. So if we look, but in a way what this reflects is also, when we're talking about not throwing away the baby with the bathwater, is about how can we modernize clinical evidence standards for homeopathy whilst still reflecting homeopathy. And so, of course, the toxicology part is part of the safety evaluation and requirement. Historical clinical use is an optional component. If there is historical clinical use, then it can be used supportively. Provings, as we've already defined within the HPCUS, is a requirement for the utility evaluation to see is there a potential signal in this substance for use in clinical practice. And what we call labeled drug discovery data, but with a much different meaning that some